your home workshop if you had the raw materials and the knowledge to do so. Yeah. Could you build as many handcuffs as Smith & Wesson does? In a year? In the same time frame? That's really a big difference between prokaryotic cells and eukaryotic cells. Prokaryotic cells are like working in your own workshop. You can do everything you need to do to build whatever it is you're building. In a eukaryotic cell, you've got all kinds of machines and all kinds of helpers to do those jobs that need to be done at the same time, so you can do it more efficiently. And today we're going to look at the structures and organelles that eukaryotic cells have in order to make it appropriate. Your objectives for this section are to be able to identify the structure and function of the organelles in a typical eukaryotic cell. A typical, those structures and functions, that is your most important objective. We are going to talk about some of the similarities and differences between plant and animal cells, and you will need to know the organelles that are found in one or the other. Many of them are found in both. Some of them are only found in plants, some are only found in animals. But your most important objective is to know these organelles or structures. So we can think of the cell as a factory. It doesn't put out handcuffs, it puts out life. And a prokaryotic cell does it like you were doing it in your workshop. It's still the same life, it's just not done quite as efficiently. In a eukaryotic cell, there are machines and things to run those machines that make it much more efficient, that allow eukaryotic cells to be larger and allow them to be multicellular organisms. We are going to skip the next two slides. These machines come in 5 different categories. The first, and the one that most of them will fall under, is assembly, transport, and storage. These organelles are going to be either building something, moving something, or storing something. And you can imagine that in a typical factory, most of the people working there are involved in one of these 3 activities. The second class would be boundaries. And we've already looked at the first boundary. We looked at the plasma membrane in the last section. That's the boundary between the outside world and the inside of the factory, inside of the life factory. Within the cell itself, there are also membranes that surround many of these organelles. Those are boundaries between inside the organelle and the cytoplasm. Support and locomotion. There's one big difference between a cell and a factory. A cell can crawl around where it needs to go, most of them anyway. So there are some organelles that are involved in moving that cell from place to place or providing that cell with its 3-dimensional structure. Control. Cells need a manager, factories need a manager. In the cell, the nucleus is going to serve as the control organelle. And then factories run on energy, cells run on energy, cells run on ATP. So we have a couple of energy transforming organelles. One that takes carbohydrates and makes ATP, mitochondria, and one that takes sunshine and makes carbohydrates, chloroplasts. So we can classify all of our organelles into one of these 5 categories. We'll start with the cytoplasm. And the cytoplasm really isn't considered an organelle, but the cytoplasm is a very important part of the cell. It's not just an empty filling space. It's not just taking up space. There's a lot of chemical reactions that go on in there, and it is continually changing and moving. We'll put it under the category of assembly, transport, and storage. It is the location of many chemical reactions. And again, it's not just a filler. It's a very dynamic and important structure within the cell. Within the cytoplasm, there is a structure called the cytoskeleton. The cytoskeleton is important for giving that cell its three-dimensional shape. If that cell moves, and many cells do, it is the cytoskeleton that gets continuously reorganized to allow that to happen, as you will see here.
The movie here that I'm about to show you is one of the most famous movies ever made in the field of cell motility. We consider it sort of the Citizen Kane of our field. This was taken by David Rogers at Vanderbilt University back in the 1950s. And what you're looking at here is a blood smear. Each of these individual round objects is a red blood cell. And this here is a white blood cell called a neutrophil. The function of the neutrophil in the immune system is to identify places where bacteria have entered the human body, to target those bacteria, move towards them, and then engulf them and eat them in order to stop an infection in its tracks. In this in vitro experiment that David performed more than 50 years ago, you see he added an individual bacterium to this culture for his neutrophil, his white blood cell, to chase. Now, as I start playing this movie, I want for you to bear in mind that this is real time. This is not speeded up, it's not time-lapse, there's no cinematographic trickery at all. And every time you cut your skin and a few bacteria enter your skin, this tiny drama is actually played out in your own body at the speed that you're about to see in this movie. So now, starting the movie, I want you to notice several things about the motion of the neutrophil. First, as the bacteria moves and the neutrophil moves after it, you can see that within a very short period of time, really a few hundred milliseconds or a few seconds at most, the cell is able to reorient all of its dynamic structures to point towards that bacterium and chase following that bacterium as it moves through this very complex environment. And then finally, when it catches up with the bacterium, it's able to engulf it. Now, the dynamic processes underlying this are, of course, obviously very important to the function of the neutrophil. But one thing that we now appreciate is that throughout the process of eukaryotic evolution, these processes have been very strongly conserved and indeed are fundamental to the life processes of most eukaryotic cells as we know them. For example, this movie shows a very different kind of cell. This is a single-celled organism called Acanthamoeba castellanii, which is an amoeba that lives in the soil. And in some senses, it's rather similar to the neutrophil in the sense that it makes a living by crawling around, finding bacteria, and then eating them as a nutritional source. We watch this we watch this amoeba under the microscope at the same sort of magnification as we watch the neutrophil. What you can see is the movement of the amoeba is actually very, very similar to the movement of the neutrophil. You see it pushing out processes, moving in different directions. In this particular case, these amoeba are starving, and so they're looking for bacteria that aren't there. But you can easily imagine if there were bacteria present in this sample, that the cell would be able to ooze its way forward, chase after the bacterium, just the same way that the neutrophil did above. Now, these cells look very similar. They're about the same size. They move at very similar rates. And if you were looking at them side by side under a microscope, you'd really have to be an expert in the field in order to tell which one was the human cell and which one was the amoeba. But these have been separated by at least 1 billion years of evolution. One thing we now appreciate is that not only is their behavior very similar, but in fact the molecular basis of this movement, the individual proteins, the way those proteins interact with each other, has also been conserved with these very different kinds of eukaryotic movement over, again, many millennia of evolution. Now, these two cells I've shown you are very similar in terms of their behavior and their movement, and they also actually have sort of a similar purpose, which is to find a bacterium and to engulf it. But there are other kinds of cells that move for different kinds of purposes. For example, in your skin, when you get a scrape, not only do bacteria enter, which can then be chased after by neutrophils, but also you somehow have to heal that wound. The cells within your skin have to crawl across the connective tissue underlying the wound so that they can fill in that gap and heal the place where you cut yourself. That kind of wound healing is primarily performed by epithelial cells. They don't chase or engulf bacteria, and yet they still have to be able to move. In some ways, the characteristics of their movement are rather different, as we can see here in a movie showing particular cells from the skin of a fish, which have been isolated essentially from a wounded spot on the skin of the animal and then put into tissue culture. And as I start this movie playing, you'll see there are some differences in the way that these cells move compared to the neutrophil or the amoeba. In particular, when these epithelial cells move, they move very straight, persistently. They're really trying to get across, close up this wound, so they move at a constant rate rather than the sort of exploratory process that you will have noticed for the other two cells. And yet, even for this very different kind of motility, which behaves differently, has different characteristics, including its persistence, occurs at a different speed, and occurs for a completely different biological purpose. Nonetheless, the molecules underlying this form of motility are still conserved between these fish skin cells, the human neutrophil, and the Acanthamoeba castellanii in the soil.
So the cytoplasm and the cytoskeleton are very dynamic structures. They belong to support, or the cytoskeleton is support and locomotion. It's constantly being reorganized if a cell is moving. It's made up of smaller proteins called microtubules and microfilaments. The nucleus. The nucleus is the control organelle. It's the control organelle because it regulates which proteins are made by the cell. Every single nucleus and every single cell of your body has all the instructions to build anything you can build. It could be used to build an entire new you. But in your big toe, the nucleus only uses the instructions for big toe cells. In your brain, the nucleus of a brain cell only uses the instructions for a brain cell. The nucleus then determines what that cell is going to be. So it is the control organelle. Much like a manager would decide which parts of whatever you're going to build in your factory you're going to work on that day. The nucleus is unusual in another way. The nucleus contains an organelle called the nucleolus. The nucleolus is part of assembly, transport, and storage. The nucleolus is used to build ribosomes, and it is the only organelle that is found within another organelle. Those ribosomes are also part of assembly, transport, and storage. Ribosomes are used to assemble proteins. They put proteins together. Ribosomes are also unusual because they are not mostly made of protein. Ribosomes are mostly made of RNA, ribosomal RNA. Ribosomes can be found in two different locations in the cell. They can be free in the cytoplasm, if they are making proteins that belong in the cytoplasm, or they can be attached to an organelle called endoplasmic reticulum. That endoplasmic reticulum is a series of networking, interconnecting tubes and tunnels where proteins are synthesized into them or other things are made. Endoplasmic reticulum comes in two different forms. The first is rough endoplasmic reticulum. It is called rough endoplasmic reticulum because the ribosomes are attached to this endoplasmic reticulum. When ribosomes are attached to the endoplasmic reticulum and you look at it under an electron microscope, it looks rough. It looks like a piece of sandpaper. Rough endoplasmic reticulum takes the proteins that the ribosomes are building, folds them, and modifies them by adding things like lipids and carbohydrates to them. Smooth endoplasmic reticulum does not have ribosomes on it. And smooth endoplasmic reticulum is involved in making complex carbohydrates and lipid molecules. Most cells have a lot of rough endoplasmic reticulum and very little smooth endoplasmic reticulum. There are some exceptions, like in the liver. So endoplasmic reticulum is also assembly, transport, and storage. And in fact, when you look at a cell with a light microscope, and what you see as cytoplasm is mostly endoplasmic reticulum. From the endoplasmic reticulum, those proteins are passed on to the Golgi apparatus. The Golgi apparatus is a packaging and distribution center. It's involved in assembly, transport, and storage. It takes the newly synthesized proteins, lipids, or complex carbohydrates from the endoplasmic reticulum, does any final modifications they need, and then packages them together with other things that are going to the same place in the cell. For instance, if you were a nuclear protein, a protein used in the nucleus, your instructions would be sent to the cytoplasm. They would be picked up by ribosomal RNA, by a ribosome, attached to the endoplasmic reticulum. You would be built in the endoplasmic reticulum, modified and folded in the endoplasmic reticulum, sent to the Golgi, and the Golgi would put you in a container called a vesicle that was going back to the nucleus with other things that were going to the nucleus. So the Golgi is the packaging and distribution center. The packages, the trucks, however you want to use an analogy, are called vesicles. You may hear Golgi apparatus, Golgi or Golgi bodies. They are all the same thing. Vacuoles are temporary storage locations. This video shows a vacuole. They're going to zoom in on it here in a minute. Here, that absorbs water from the environment, and when it is full, it squeezes the water back out. It's called a contractile vacuole. Vacuoles then would also be part of assembly, transport, and storage. They are temporary storage locations. Vacuoles are a difference between plants and animals. Plants have large vacuoles. Animal cells tend not to have vacuoles. If they do have vacuoles, they are small vacuoles. Lysosomes. Lysosomes we're going to put in assembly, transport, and storage, but their job is to recycle for the cell. They contain digestive enzymes. So if you are done with a protein, or if that protein is no longer working, you send it to the lysosome. The lysosome will break it down into amino acids, and you can use those amino acids to build new proteins. Lysosomes also play a role in development. As we are developing, some cells need to die by a process called apoptosis, and lysosomes are very important in that process. If you happen to be a single-celled organism, a lysosome is your digestive system. Those digestive enzymes need to be kept within that lysosome membrane because they would digest anything in the cell if they were not separated from the cell. Lysosomes are also a difference between plant and animal cells. Animal cells have lysosomes, plant cells do not. Centrioles belong to a class of molecules called motor proteins. Centrioles are used to move DNA during cell division. But there are a lot of motor proteins and a lot of things going on in the cell.
Let me take you here into the interior of a squid giant axon seen in real time through a video microscope, very much like the original experiments of Bob Allen, Scott Brady, and Ray Lassick. And what you can see here is a tremendous amount of motility, all kinds of small organelles which are traveling between the nerve cell body and the distal nerve terminal. And these long snake-like things here are mitochondria that are also moving through this very dense cytoplasm. Indeed, if you turn a microscope to any eukaryotic cell, you will see lots of intracellular motility like this. Here's another prominent example of intracellular motility, the process of cell division. Here the DNA in green, the microtubules in red. The DNA congresses to the middle, and then the physical separation of the sister chromatids to equally partition the genetic material. Now, all of this fantastic intracellular motility is driven by biological machines called molecular motors. And that is going to be a major subject of these lectures. And just to get you in the right spirit for thinking about these molecular machines, I'd like to show you an animation that was developed by Ex Vivo in conjunction with Harvard and the Howard Hughes Medical Institute that takes you into the interior of the cell to show you a little bit about what these machines might be doing.
vesicle from the Golgi. Mitochondria we'll talk about in a moment. Here's the centriole.
What I hope you can appreciate from that remarkable video is that the cell interior is packed with interesting machines. Maybe they don't work exactly as they're shown in the video, but the spirit of the video shows that the inside of cells is very complicated. There are lots of molecular machines that are working together to create all of this complex cell behavior that makes cells function and execute a number of complex behaviors and abilities.
Okay, so centrioles are used during reproduction. They move DNA. Centrioles are another difference between plant and animal cells. Centrioles are found in animal cells, not in plant cells. Plant cells still have to move DNA during division, but they don't use centrioles to do it. Mitochondria, saw one on the video. Mitochondria belong to the energy transformers. Mitochondria provide ATP to the cell by burning carbohydrates. They have a highly folded inner membrane. Whenever you see highly folded in biology, it's because you can get a lot of stuff in a small space. So if there's a lot of it, it's probably important. The inner membrane is very important in what mitochondria do. Also, this is not a difference between plant and animal cells. Both plant and animal cells have mitochondria. The mitochondria do the same thing, convert carbohydrates to ATP. This organelle looks very similar to mitochondria. It is a chloroplast. A chloroplast is also an energy transformer. Whoops, doesn't have a card. Chloroplasts take sunlight energy and use it to build carbohydrates. So if you have a chloroplast, you can make your own carbohydrates, send those carbohydrates to the mitochondria, and make ATP. Chloroplasts are a difference between plant and animal cells. Plant cells have chloroplasts, animal cells do not. Animals have to get their carbohydrates from somewhere else. A cell wall is actually on the outside of a plasma membrane. A cell wall is also a difference between plant and animal cells. Plant cells have cell walls for extra support. Animal cells do not. Cilia and flagella. Are extensions of the plasma membrane. A flagella is a long whip-like structure. It is used to move a cell from place to place. Most cells only have one flagella. There are a few microorganisms that have two flagella. In humans, there is only one cell that has a flagella, and only approximately 50% of the human population has that cell. Cilia are more numerous, shorter extensions of the plasma membrane. If you are a single-celled organism, you might use cilia to move around. But in a multicellular organism, cilia are going to be used to move materials across the surface of the cell. For example, we have a large number of cilia in our respiratory system, and they beat in such a way to move mucus from the lower parts of our respiratory system to the upper parts of our respiratory system, so we can get rid of it in the form of a cough, a sneeze, or even swallow it. So we're going to call these part of assembly, transport, and storage. They either move things over the cell or move the cells themselves. Cilia are smaller and much more numerous than flagella. This one we're going to skip. Animal cells have centrioles. Small vacuoles, if they have them at all. We're going to consider vacuoles a difference between plant and animal. And not listed, but should be. Animal cells have lysosomes. Plant cells have chloroplasts. Large vacuoles. In fact, if you see a cell with vacuoles, you can assume it's a plant cell. And cell walls. So animal cells, centrioles, no vacuoles, and lysosomes. Plant cells, chloroplast, vacuoles, and cell walls. So we started out this by saying that cells could be thought of like a factory. And I said the factory would be putting out a product we'd call life. In reality, to put out life, you have to build proteins. So you can also think of a cell as a protein building factory. The nucleus contains instructions, and it makes the ribosomes that can put those instructions into work. Those ribosomes turn the instructions into proteins. The ER modifies those proteins. The Golgi delivers those proteins where they need to go. And mitochondria provide the energy so that they can do their job, giving us a very efficient life factory. Same kind of work goes on in a prokaryotic cell, but it doesn't have all these machines and workers to do it. So eukaryotic cells contain membrane-bound organelles in the cytoplasm that perform cell functions. Ribosomes are the site of protein synthesis. Mitochondria are called the powerhouse of the cell because they provide ATP to run the things that need to run. And plant and animal cells contain many of the same organelles. Others are unique to either plant or animal cells. Plant cells, chloroplast, cell walls, vacuoles. Animal cells, centrioles, no vacuoles, and lysosomes. Any questions? I will be putting up the next 5 assignments as soon as I sit down, pretty much.